It's a great honor for me and a pleasure to welcome this year's Pioneer Award recipient and keynote speaker, Professor Catherine Boileau. Catherine is not only a fantastic researcher, she is also a wonderful person. Professor Boileau has transformed our understanding of familial hypercholesterolemia through her pioneering work in biochemistry and genetics. In 2018, she received the Lefoulon-Delalande Foundation Scientific Grand Prize for her 2003 discovery of the role of PCSK9 in cholesterol metabolism. In 1998, Professor Boileau founded the CHOPIN project, a national research network dedicated to the identification of new genes implicated in FH. Building on this work, Catherine and her team followed French families affected by FH and identified a causal association within a region of chromosome 1, previously known as NARC1, but now as PCSK9, in these families and in the kindreds of FH families in other countries. These discoveries paved the way for the development of PCSK9 inhibitors, a revolutionary new therapeutic option for those with FH and coronary artery disease. Professor Boileau has performed pioneering research for over 35 years and has authored over 300 international publications. She has received numerous awards and honors, including serving as the first president of the Scientific Advisory Board of the French Foundation of Rare Diseases, and has been recognized as a Chevalier of the French Légion d'honneur. That's a knight. Can the woman be a knight? Is it a knightress, I wonder? A knight of the French Legion of Honor. Professor Boileau has led research teams at INSERM and now heads the structural cardiovascular diseases team at INSERM Laboratory for Vascular Science. Professor Boileau is the chair of the genetics department at Bichat Hospital in Paris and teaches at the medical school of Paris Diderot University. She's currently on the governing board of the Paris University Hospitals and is the director of research of the Northern University Hospitals in Paris. Please join me in welcoming this year's Pioneer Award recipient, Professor Catherine Boileau.
Thank you. Thank you very much.
Dear Dr. Boileau, thank you for applying your great intellect and dedicating so much of your time and effort to the pioneering scientific work that you have done. I would like to present the 2019 FH Pioneer Award to you on behalf of all the individuals with FH.
Thank you. Thank you very much.
The group hug.
Sorry.
Come on, because I'm shorter.
This is, okay. This is a nice picture, okay. First of all, I want to thank you, Catherine, the FH Foundation, the board, the Scientific Advisory Board for choosing me as recipient of this wonderful honor this year. I am thankful, but I'm also very proud because I am getting into a group of outstanding scientists, far better than I am, I think, who received this award in the previous years. I am very humbled, and I am moved, and I won't go any further on that subject. I dedicate this award to you, Catherine, Kate, and your husband and your children, and all of you. It's always been my purpose, through my years of work, to try to be helpful. I do a lot of diagnosis. I'm a biologist, I'm not a clinician, but I'm happy that some help comes out of all this. And to end my talk, I want to pay tribute. This award was possible only because there were other people. You never do research by yourself. And I want to pay tribute to 3 people. The first one is Nabil Seidah. Nabil Seidah cloned NARC and agreed to let go of the name of his child and to rename it PCSK9. The other person, the second person, is Marianne Abifadel. Her commitment in doing all the genetic work was incredibly good, and it involved a lot of self-sacrifice, and for her, I'm very happy we made it. And I want to pay tribute to a third person who's a member of your scientific advisory board, Paul Hopkins. Research is a cutthroat field, and our strongest competitors were headed by Paul, and we just made it by a margin before he did. But he needs to be recognized because with his work, not only did he replicate our results, but he also identified the D374Y mutation in PCSK9, which has been so helpful in helping develop the inhibitors. So thank you again. Thank you. Thank you, Anne. Thank you, Josh.
And now we'll
you get a hug.
The privilege.
You also get a
hug.
hug.
Oh, excuse me.
You get a hug.
And we even practiced, but.
Yeah.
And now we'll have the privilege of hearing from Dr. Boileau.
After Kate's talk, I feel terrible because I'm just a scientist. You know, I don't have a very interesting life as compared to hers and her husband, and she has wonderful children, and I hope for them that we will all be able to do better than we did up till now. My talk had no title, as you've noticed in the program. So the title is Variations on the Theme of FH Variability. And this is a metaphor. And for those of you who wondered, what is this title? What does this mean? Actually, I was thinking, oh, well, I have disclosures, so that'll get that out of the way. I was thinking more of the great musical variations and variations in theme. For those of you who are into music, be it classic or non-classic, a piece of music has a theme, which is the melody. And then the composer improvises on variations on the melody. And you've heard of this in jazz very often, but you don't think of it in a classical way. And the variation is music that is similar to the theme, but is different. And I think this is an excellent metaphor for the fact that FH, of course, is a disease, but each carrier has a variation on the theme of FH. And for those of you who are familiar, some variations that are very famous, the Bach-Goldberg variations and Mozart's. And I think, thinking of it, I think Twinkle, Twinkle, Little Star is a variation, has many variations, and I think your children are variations on Twinkle, Twinkle, Little Star. So, great musical variations, great variations in FH. And actually, when you think of life, there are variations everywhere. Variations on themes that are different. You have the various finches here, the ladybugs, the shells, the carnations, you have the rabbits, and of course you have humanity, the human species. We are variations on the theme of humanity. In the case of FH, the problem is, what is variability? Why is one form of FH disease different from one individual to another? So what does it due to? It's due to something that we geneticists have known for a long time and we all agree on, is the fact that the phenotype, when you think of the phenotype, and this would be FH, the disease, is due to a combination of various effects. The effects, of course, are genetics, and there's the mutation in the gene, which triggers the disease. But then this mutation is within one gene, knowing that the body has over 25,000 genes. So you can understand that depending on the genetic background, the expression of the mutation will be different. And then there are other effects, which are the environmental effects. And you don't always think of it, but we are, all of us are subject to environmental effects from the moment we are a fetus, and we have intrauterine environment. And then afterwards, after birth, we have our own environment directly through various food, various drugs, but also our behavior, our age, our sex, the exercise we do, what we eat, but also what we smoke, the diseases we have, that all contribute to some kind of metabolic modifications. And we now know that these metabolic modifications are able to modify gene expression. That this is what we call epigenetics. So this is complicated, but this explains why we're all so different, and of course FH patients are different. Now, how do we detect the drivers of these differences? Well, actually, we have several. I'm sorry, here it is. I'm sort of getting, I think I should shoot over there. So one way of doing it is doing population studies. What you have here are two studies that were done in the mid-90s in Chinese patients, either from mainland China or from Canada, who all carried identical mutations within the LDL receptor. What you see on the left-hand side of this slide is the mean LDL cholesterol level in the mainland China population. And I think you'll agree with me that this value, if you're not familiar with it, this is approximately 166 milligrams per deciliter, is very much lower than the one observed in Canada for the Chinese carriers of an identical mutation who had over 288 milligrams per deciliter. And there's no doubt about it. What is the difference? They're all Chinese, they all have the same mutation, but, and I think you'll understand this, the patients in China come from rural regions out of Nanjing, and their food at the time, especially in the mid-1990s, was mostly starches like rice or noodles, some vegetables, very low amounts of meat or fish, and absolutely no dessert, sometimes fresh fruit, and that was it. Conversely, the British Columbian Chinese immigrants had a far different diet with whatever you think of the diet pyramid for the Western type of nutrition appears here, and was probably associated, in the case of the immigrants, with some very heavy Chinese holiday food, as well as our regular intake of junk food, let's face it. I'll just call it junk, but I do love it, unfortunately, too. Okay, now another aspect is variability within a family, an FH family, and Eric is here, who published this paper many years ago, and he can even tell you more than I can about this large family from the Netherlands. The Netherlands is a wonderful country because they have these registries of life, of death, of disease, and so they were able to build up this whole pedigree that goes back to the 18th century, to the founders, and these founders gave, let's say, birth to 3 branches of this family that has over 400 descendants, and 118 of them had heterozygous FH. And in this family segregates a mutation that is well known, that it was first described in southern Africa, and is called Afrikaner 2. What I'd like to show you is this: the evolution, the mortality rate between 1840s and the 1990s for women and men in this family. And as you see, through the years, mortality rates were elevated until the the early 1930s, and then they went back down, mostly thanks to statins. Another aspect is the survival that was is the survival that was looked at within the various family branches. So you have branches A, B, and C. What you have here is the total number of members within each branch of the family, and every time one of the members dies, you have a step down. So what you see clearly is that in the A branch, which appears here in red, individuals die earlier as compared to the branches B and C, where individuals really live much longer. And what you have here actually is the typical aspect of an environmental effect and the genetics effect. Now, the question after that is, how do you define FH variability? What is different from one FH subject to another? You can either define it with actually with lipid levels. What you have here is a molecular picture of the variants that are gain-of-function variants today, known to be located within the PCSK9 gene. And I'd like to concentrate on one of these mutations, which is the S127R mutation that we described many years ago. This actually, for us, is a French founder mutation. We now have 13 families with this mutation. And so we are able to study carriers of an identical mutation and see what are their, let's say, various parameters, clinical parameters and biological parameters. And as you will see here, the age before treatment varies greatly. Great variability. And of course, after treatment, this is to give you an idea of the age at which these subjects were included within the study. Now, their BMI, the normal BMI should be around lower than 30, and 24, even 25. And you see that we have overweight patients that carry these mutations. And when you look at the lipid levels, we use something that is called the multiple of the median, which allows to compare biological values that are different between sexes and differ between age groups. So we use what is called a MOM, and so this enables us to compare all the values obtained within the carriers of this mutation. What you have here are their triglycerides, and you see that here, when you have a MOM of 1, it means that your level is the exact identical to what should you expect for your category of age and sex. Whereas when you're above that, you have much elevated, more or less elevated values. So you see that the triglycerides varied greatly, and you didn't have a very pure hypercholesterolemic phenotype. HDL-C values also varied, but look at the LDL cholesterol values. The huge variability. The same mutation. So you would say they're all FH patients, but actually when you look at their LDL levels, they're not identical. No one is identical. Now, another way of saying this is also looking at these patients as members of various families. And again, what you see is even in a given family, although there seems to be a clustering around a median sort of value, you do have outliers, and this is important to know. So you can define variability by looking at your lipid levels, or you can also use other variables, and I'll go back to that afterwards. Now, the question will be, how do we find the drivers of this variability? This is really the main goal for the years ahead. As I told you, I think my generation of scientists, we identified the initiators. The next generation needs to find, well, I'm still part of the next generation, I think. So I'm helping the next generation find the modifiers of so the drivers of this variability. So what are the possible drivers? And that's where the headache comes in. Because they cannot either be within the lipid metabolism. And what you have here is a picture of something that all medical students hate. And as a teacher of clinical biology, I can tell you that when I go into my lipid metabolism, all of a sudden they all fall asleep. And so it's only after working 35 years in the field that you feel comfortable with something like this. So anyhow, this is just to show you, oh, I'm sorry, I'm not very good with this. This is just to show you the whole complexity of lipid metabolism. And when we're talking about elevated LDL, here they are. Here they are, but then you've got arrows everywhere, meaning that if you dysregulate something around here, you're going to have effects all the way to the other end of the metabolism. So the drivers are somewhere in there. Well, how do we find them? Another way to look at the possible modifiers is to look not at the lipid metabolism, but at the consequence, which is atherosclerosis. And for those of you who are not familiar with the process, this is what a normal aorta would look like, and this is what it looks like when it's explanted. This is the fatty streak with moderate atherosclerosis. Here it is, seen from the outside, a little lovely picture. And this is severe atherosclerosis. And you have a fibrolipidic lesion. And as you can see, the vessel lumen is getting narrower and narrower, and that explains the complications of the disease. No, so how does this happen? Well, again, you're going to have a headache. This is what happens. And this is just, you know, a cartoon that sort of, you know, is an abstract of what is happening, because this is a very complex metabolism and process that involves many cells, the endothelial cells, the smooth muscle cells, the macrophage cells, and then you have oxygen species, you have cytokines. I mean, it's so busy in there that any modification within one of the actors can have an impact on the other actors. So again, many possible drivers when you look at the drivers of atherosclerosis, and I think you will agree with me that the word cornucopia of drivers is the proper word. So how do we find the drivers within this mass of possible drivers? Well, I think there are two ways to do it, and I'm very keen on the first way because I've used it for many years, and I think it really is something that has a lot of power when you do research, and I can only emphasize that the importance of doing family studies when you do research. So what do we need? We need big families. The question in FH is easy because it's a family disease. The only thing is, in Western countries, we don't have many children anymore. And so if you want to have a very large family, because these family designs need large families because you need a large number of heterozygous FH patients or homozygous patients and their quote-unquote healthy SIBs. So one of these families was a family that was published many years ago by Helen Hobbs. It's a family that came from Puerto Rico, and I'd like you to look closely at the family. I'm sorry for those in the back, but the half-black symbols are the heterozygous carriers, and the proband, the index case, is this homozygous subject. And the arrow points to his mother. His mother is what we call a case of incomplete penetrance or skipped generation. Why? Meaning because this woman has a father who displays familial hypercholesterolemia at the heterozygous level. She gave birth with her affected heterozygous FH husband to a child that is a homozygous FH, meaning she's a carrier. She's an obligate carrier of her father's mutation. And lo and behold, when you find the mutation, indeed, she does carry it. This is the mutation that was identified. But look at the family. Look at the family. All the subjects that have blackened symbols do carry the mutation. And the only ones that appear here with the asterisks, the pink asterisks, are the patients, the subjects in this family who carry the mutation but have no FH phenotype. And having a family like this is a treasure, is an incredible treasure, because this is where you can find, actually, you really want to find out what these subjects, these relatives, have in their makeup, their genetic makeup, as well as their environmental makeup, that makes it for them possible to have a bona fide lipid elevating mutation and no expression at all of the disease. At the time, Helen Hobbs had demonstrated that this is probably a modifier gene that had an autosomal dominant mode of inheritance, meaning that carriers only needed one copy of the variant for it to have an effect. Another such family is one that was reported by Aaron Leitersdorf, also a certain number of years ago. It's a large Muslim Arab family living in Israel. You have it here, and all the individuals in red, the red symbols, are homozygous subjects, and you will agree with me that there are many within a single family, but this is easily explained for families in the Middle East, in Israel, as well as Jordan, Syria, Lebanon, where there is a high level of consanguineous weddings. And so what you have here are numerous homozygous subjects and, of course, numerous heterozygous FH subjects. In this family segregates a mutation that is well known. It's the JD mutation. JD is the historic mutation that was carried in the families that Chadourian studied, and in which he described the disease, saying that it was an inherited autosomal dominant disease. And the JD mutation is also the mutation that was present in the fibroblasts that Brown and Goldstein studied and showed all the mechanism of LDL receptor and LDL complex endocytosis. And it was only 10 years later, after the description by Brown and Goldstein of the mechanism, that Davis and collaborators showed that at the molecular level that this is a single nucleotide substitution. So that's for the historical part. Now, what is interesting is that in this family, like the Puerto Rican family, you have a certain number of subjects who have the disease, who should have the disease, but actually have very lower levels of LDL cholesterol than expected. And looking at all subjects that have values under 150 milligrams per deciliter, then these subjects appear with the blue asterisks. What became evident to Aaron Leitersdorf was that there was, again, a modifier gene, which this time segregated as an autosomal recessive, meaning that subjects needed 2 copies of the variant for it to have an effect. Now, what he did was, with this very large family, he did family analysis, genome-wide family analysis, meaning that using markers that we have hundreds of thousands of markers that really reference regions within the genome. So he used these markers to try to identify in carriers and non-carriers a region that was only present in carriers and that had been inherited. And this region is here. It appears here. Actually, what you have are just some branches of the family, and the smallest area of complete sharing between branches is here. It's a region on chromosome 13, at the tip of chromosome 13. And that's the story that ended within the paper that was published at the time. And for the purpose of my talk today, I did a quick query on what we know now of that area using the genome browser for chromosome 13. These are the markers that were used by the authors. And here are the genes that are now located within this region. And I can tell you, you can send a message to Israel. They better have a good look at these genes, because those are excellent candidate genes for a modifier. Okay, that was not my subliminal message. Now, how do we find the drivers? Second method, that's the population study, or what we do as a population studies, what we did in my lab, and I'll just go quickly through them. It's not at all the kind of studies that will be addressed probably tomorrow by Catherine. It's just another way of looking for modifiers, and I'll show you when you don't have big numbers of subjects available. So you decide to define your modifier gene on its effect. So either the effect is on the lipid phenotype, and when you look at the distribution of LDL cholesterol levels in the general population, or PCSK9 blood levels, you see that the distribution is what we call a normal, a bell-shaped curved distribution. You can do this with lipid values, but you can also do this with a marker of coronary artery disease, which is the coronary artery calcium score, for which we have values in women and men. And these values appear here. You have distribution of the scores depending on the age of the patients, and here you have the number of patients who have the various scores. And again, if you look closely, you do have bell-shaped curve distributions, both in men and women for this parameter. So our strategy is to use what we call the extreme phenotype sampling strategy, meaning that what we do is study patients from the ends of these distribution, the top end and the bottom end in men and in women, thinking that these patients, these FH patients, are enriched for variance, drivers of variability that either lower or raise the effect of the parameter on either lipid levels or coronary artery disease. And doing this, what we do actually is do either just extreme phenotype sampling and have a benign group versus a severe group in which we do GWAS analysis or whole exome sequencing. And we also do a little family studies. Why? Because there's much more power. There's always much more power if you can do relatives. And so we do SIB pairs. And what we do is that we take SIB pairs with the highest values, and what we do is that we try to look for concordant SIB pairs, meaning that the pairs of SIBs have the same manifestations of the disease, or discordant pairs. And for this, we do, again, genome-wide linkage and another kind of GWAS that is done in relatives. And what we do is do this. We did this a couple of years ago in another disease on which I've worked a lot these past years. So the design is basically the same. This is what I just showed you. And the results we obtained identified a certain number of loci with each method. And what we did is we cross-mapped our results, and where the intersection is between all the four strategies, we have a locus that is a possible driver of the modification. And so you see that with this method, we were able to identify 11 regions within the genome that carry modifiers. And I'd like to show you just closely this region for modifier 1, and this is what our results look like. This is the GWAS results, this is the SIB1 part of the SIB pair results, and this is the linkage in the SIB pairs. And we define a minimal region in which the modifier is located, and lo and behold, a very interesting gene as far as an aortic disorder is concerned. So this is what we hope to get when we do an EPS method within the LDL, the FH disease, with patients carrying an LDL receptor gene mutation. I'm sorry if I'm showing you more my back than my front here for people on this side of the room. Now, what do we hope to gain by all this? Well, I think you will agree with me that using whatever method we use, what we hope to get is more, of course, more understanding of the various steps that lead to atherogenesis and to the complications of the disease. And we also hope to identify within families the atherogenic drivers. And so we need an atherogenic driver profiling that could be obtained. And having these results would have two consequences. First, by defining drivers, we can define targets to develop new genes, like PCSK9 was a target for new therapies. I'm sorry, not defining new genes, but defining new therapies. And another thing that will be possible is that we can profile patients and know who will be at risk of a more severe disease or a less severe disease, and thus probably with these new drugs and even with the ones we already have, give drugs that target really the atherogenic drivers and that target the proper patients within the FH populations that need to be followed. Now, if I go back to my metaphor, my original metaphor, using all these methods, maybe, probably not, but I don't think we will decipher or at least have the complete score of Mozart's variation on Twinkle, Twinkle, Little Star. But if we at least get the easy variation, the way to the score for the easy score, maybe we'll be able to help FH people down the line. Thank you. Thank you.
Wonderful.
I took a lot of time.
I think
that was fantastic talk, and we will take questions from the audience in a few minutes, so think about them. But I get to use the host prerogative and ask the first question. You know, as a scientist who's after discovery, it's always intriguing to me about whether there was an aha moment, or you know, when you hit the baseball out of the park in the middle of the night, you know, in the ninth inning, what was that moment like, and how did you celebrate? And tell us about discovery PCSK9.
Actually, it was to tell you the truth, it was not a ha-ha moment. It was just one of the numerous. We had done so much sequencing in that area for such a long time, and we had found the first variants, and they were interesting. They were really among the good variants we had identified. And it was only when we started having, through collaboration with Nabil Seidah, the first functional test, that the ha-ha moment was there. And then we really were happy. That's true. We were really happy to be there. And I think, as any researcher will tell you, the aha moment is when the publication is accepted in an exceptional journal. That's when you drink heavily, and we drank. We drank. Let's face it, wine is good in France, and champagne is even better.
I have another couple questions, but one of them is, you know, you showed a lot of pedigrees, families, yeah. You know, and at the FH Foundation, we just, you know, the summit, we heard an incredible example of a family, yeah. And the role that families, yeah. Play, and you know, sometimes you call them research subjects. Yeah. They're not really subjects; they're participants. And yeah. So, tell us about the role that the FH community played, and I know you might have some funny anecdotes potentially, or so.
Well, there was no French FH community at the time. It's only been recently that there is really a strong FH foundation, the ANET, which was on my first slide, and they're a great work, and they're doing a great work. So at the time, what I did was I set up a research network, and there were 5 centers where lipid clinics had families, and we were able to enlarge families. And some of the families, it was really hard because branches of family, let's face it, all families have their own history, okay? And then you have, I'm talking about French families, okay? We are in a sort of, at the northern part of France, you could say this could be America. But the southern part certainly isn't America. It's Sicily.
Thank God.
And so you have to sort of navigate the culture. But I remember 2 families, notably one in which there was a great aunt who was in touch with her sister-in-law. And she really got everybody together for a party. We call it a rally or something like that. I mean, the family gets together so the younger generation can meet their cousins. And on that day, there was first the sampling, and then the champagne. And so that's how we got probably one of the biggest families we were working with. So, no, we were very fortunate, I think, in, and I really want to emphasize the importance of helping, participating. Things are very different. Here we're 25 years later, the social network exists. I mean, there are various ways of getting in touch with people. Things are very different, but basically it's still the same. I think you truly have more power, or a different kind of power, when you're studying families, and not only because for a researcher you have more power, but I think for these families you have a way of educating them and getting them to participate, but it's not always easy.
So do you think we're going to find more dominant FH genes?
Oh, that's a hard one. I tackled that one last year with you. We're missing heritability. There's heritability that's missing. So where is it? Where is it? And one of the answers to that question probably will be switching from whole exome sequencing to whole genome sequencing. We've missed it. We've missed mutations that are probably in known genes.
Yes.
So we hopefully hope that there will be more in there. Actually, not more genes, but more ways of identifying. I'm not sure there are a lot of dominant genes out there.
No, I agree. Yeah.
I'm lucky enough to be sitting on the stage with two women who have been so important in FH care, and we're led obviously by Catherine. And I've looked at the program before I came up here and realized we have 14 of our 25 speakers this year are women, which is
hooray.
We should
hooray.
pat ourselves on the back. But you know, so science, I'm not sure how easy it was back in France those years ago, but we have a lot of junior investigators in the audience and women, and what advice do you have for them?
Okay, male or female doesn't make any difference. It's just the number of hours you stay in the lab and working. When it'll start making a difference is on the boards. It's getting on the boards. And I don't think it's just a question of discrimination. It's a question of gender and the way we approach commitments. I'm sorry, this is leaving me. We women tend to be overachievers, and don't listen to this. Men, don't listen to this. Meaning that we will say, yes, okay, I'll go on board when we're offered. I will go on board if I have the time to do it properly. I think a lot of our male colleagues will go on board, will do the job, but they won't be finicky about all the little aspects. And I'm not sure it's necessary to be finicky, but we were just brought up this way, at least in my generation. So there is the gender gap, it's true. There is cultural, it's historic, it's changing a lot because there are a lot of women within the research community, and I think this is a place where the gender gap is the lowest, to tell you the truth.
All right, I don't know if I can use this. So thank you so much for that amazing talk. And I really have been thinking a lot, and I failed to say it in my first remarks, about the fact that we're going to know more and more about everyone and their genetic vulnerabilities. And I think that's one of the things we need to think responsibly about. If we, you know, there's the upside that we've been talking about, which is understanding the drivers of pathology, and then the opportunity to develop therapies to intervene at that particular place in the metabolic disorder, et cetera. But you know, I read in the Wall Street Journal, I think a week ago, that it's estimated that there'll be over 13 million people who've had their whole genome sequenced in the next 4 years. And I think one of the topics that we're looking at, whether it's genetic or otherwise, as we begin to be able to predict risk, is I'm curious what responsibility you all feel as researchers to be a part of the conversation about if we ask people to participate in research and learn their vulnerabilities, how are we going to take care of them in the larger questions that surround living with that condition? So I'd love to hear all your thoughts.
Well, I think you can also answer that question. We can all, as researchers, answer that question or try to answer it. Unfortunately, technology is going quicker than knowledge, meaning that we can have whole genomes sequenced, but we still don't know what is the meaning of a good part of what we have. And so that's one aspect, and basic knowledge has to catch up. But that's okay. I mean, this is the normal evolution of science. The problem is, I think, is that people are getting their DNA sequenced without being enough informed of the information that's going to come out. Because unfortunately, the only information we're capable of seeing is the bad news. We're incapable of telling you you're going to have a healthy life. We're only capable of saying, oh, wow, you have a risk of having colon cancer, breast cancer. So that is really heavy, very heavy. And it's very different. This is what comes out when you have your whole genome sequenced. And in a way, you cannot predict. Let's face it. We do not have the information to predict. What we have is the information to say there is a risk, you need to be carefully followed up. And that's good information. But that means that you need to have a practitioner, a GP, a specialist, a cardiologist, an oncologist, who has the time to explain all this. And that has consequence, is important in terms of the amount of medical time or nurse time that is necessary to explain results. So I don't have a quick answer to that question. It is very worrisome, and there's something in the French bioethic laws that says that you need to have informed consent. We cannot go online in France and get DNA sequenced. That's not possible. We go online and have it sequenced in the States, if you want it, okay? So the French bioethic laws, for the time being, say that you cannot have your DNA sequenced except for medical reasons. And then there's a whole way of doing this. But the bioethic laws also say that you have the right not to know, not to know. And I think that's something we should all think about. Maybe not all of us want to know if we have a risk of being Alzheimer. Alzheimer you cannot treat, but colon cancer you can follow. I mean, you can prevent, more or less. So this is really something that we all need to think about as a community and worldwide, to tell you the truth. So I don't have an answer, I just have questions.
I completely agree with that, right? And we have the same kind of laws in Denmark, right? That you have to have informed consent, also the right not to know. So I think, you know, as always, this is an American phenomenon, and Europe is just a little behind, right? Because I don't think many people actually want to have their genome sequenced. Why would you really want that?
Well, it depends. Yeah. It depends. You know, you get it for Christmas. It's not that expensive.
I'm probably E4. I don't want to know that.
Well, I wouldn't, but then I'm old. I'm old. I mean, we also have bioethic laws because, you know, no discrimination depending on your genetics, okay? And then you go on Facebook. Guess what? I just had my genetic results. Here is my mutation. I mean, it's a generational thing, too. I'm not sure. We need to have young people. Are there young people in the room?
Oh, yeah.
I think they're the ones who should answer these questions, not the people with the white hair. Knowledge is also power. And when you're empowered, to take care of yourself in your life. That might be an important aspect. I've always been told by my colleagues from the French network that actually you don't need to have the FH mutation to make a diagnosis of FH. You only need to have a good clinical exam, someone who knows what xanthomas are, and some biological tests. But if you have the mutation, there's better compliance. There's a whole better way people take better care of themselves. So maybe knowledge is power.
Yeah.
So I think this will be, maybe this will be our last question. Go ahead.
Okay. I am Marcos Lima from Venezuela. Congratulations for your award and very nice talk. Congratulations. I have two questions. The first one is, what is the contribution of the new genes, for example, sortilin genes, in the variability in LDL levels in patients with FH? And the second question is, I am an endocrinologist, and I would like to know, what is there evidence about endocrine disorders, the influence of endocrine disorders in LDL levels in patients with FH? Probably with insulin resistance, hypothyroidism in that patients? Thank you.
Okay, that's Anne would probably be a better person to answer that, because what you have, it's not because you have FH that you are free of modern diseases, of modern diseases of the Western society. Meaning if you have diabetes, well, you will have diabetes, and your effect on the LDL will not be only on the LDL, you'll have an effect on the triglycerides. You'll have a more complex effect. So I would say that I agree with you, the whole picture can be different if you're looking at an adult patient who has an elevated BMI, who has type 2 diabetes, and, or a menopausal, post-menopausal woman who will have, it is well known that the estrogens have an effect on cholesterol levels. And very often, you can, pretty often you can see that all of a sudden, post-menopausal, you have a very high raise of total cholesterol and LDL cholesterol in a certain proportion of women. And so some FH women probably will be revealed only post-menopausal.
I think we have some of the world's experts on polygenic risk scores here, and I think the data is pretty clear that if you have a high polygenic score plus an FH causal mutation, your outcomes are going to be worse, and if you have a low polygenic score.
Exactly. But then that's just additive.
Exactly.
Sort of. It's additive. Or, well, we can discuss about synergistic or additive.
Right. So I think we should thank Dr. Boileau for...
Thank you. Thank you.